Spider life defined. Spiders order Araneae are air-breathing arthropods that have 8 legs, Chalcedoe with fangs generally able to inject venom, and spinnerets that extrude silk. They are the largest order of arachnids and rank 7th in total species diversity among all orders of organisms. Spiders are found worldwide on every continent except for Antarctica and have become established in nearly every land habitat. As of August 2022, 50,356 spider species in 132 families have been recorded by taxonomists. However, there has been debate among scientists about how families should be classified, with over 20 different classifications proposed since 1900. Anatomically, spiders, as with all arachnids, differ from other arthropods in that the usual body segments are fused into 2 stegmata, the cephalothorax or prosoma and the opisthosoma or abdomen, and joined by a small cylindrical pedicel. However, as there is currently neither paleontological nor embryological evidence that spiders ever had a separate thorax-like division, there exists an argument against the validity of the term cephalothorax, which means fused cephalon head and the thorax. Similarly, arguments can be formed against use of the term abdomen, as the opisthosoma of all spiders contains a heart and respiratory organs, organs atypical of an abdomen. Unlike insects, spiders do not have antennae. In all except the most primitive group, the mesothelid spiders have the most centralized nervous systems of all arthropods, as all their ganglia are fused into one mass in the cephalothorax. Unlike most arthropods, spiders have no extensor muscles in their limbs and instead extend them by hydraulic pressure. Their abdomens bear appendages that have been modified into spinnerets that extrude silk from up to 6 types of glands. Spider webs vary widely in size, shape, and the amount of sticky thread used. It now appears that the spiral orb web may be one of the earliest forms, and spiders that produce tangled cobwebs are more abundant and diverse than orb weaver spiders. Spider-like arachnids with silk-producing spigots appeared in the Devonian period, about 386 million years ago, but these animals apparently lack spinnerets. True spiders have been found in Carboniferous rocks from 318 to 299 million years ago, and are very similar to the most primitive surviving suborder, the Mesotheli. The main groups of modern spiders, Mydalomorphae and Araenomorphae, first appeared in the Triassic period, before 200 million years ago. The species Bagheera kaplingi was described as herbivorous in 2008, but all other known species are predators, mostly preying on insects and on other spiders, although a few large species also take birds and lizards. It is estimated that the world's 25 million tons of spiders kill 400,800 million tons of prey per year. Spiders use a wide range of strategies to capture prey, trapping it in sticky webs, lassoing it with sticky bolas, mimicking the prey to avoid detection, or running it down. Most detect prey mainly by sensing vibrations, but the active hunters have acute vision, and hunters of the genus Portia show signs of intelligence in their choice of tactics and ability to develop new ones. Spiders' guts are too narrow to take solids, so they liquefy their food by flooding it with digestive enzymes. They also grind food with the bases of their pedipalps, as arachnids do not have the mandibles that crustaceans and insects have. To avoid being eaten by the females, which are typically much larger, male spiders identify themselves to potential mates by a variety of complex courtship rituals. Males of most species survive a few matings, limited mainly by their short lifespans. Females weave silk egg cases, each of which may contain hundreds of eggs. Females of many species care for their young, for example, by carrying them around or by sharing food with them. A minority of species are social, building communal webs that may house anywhere from a few to 50,000 individuals. Social behavior ranges from precarious toleration, as in the widow spiders, to cooperative hunting and food sharing. Although most spiders live for at most 2 years, tarantulas and other mygalomorph spiders can live up to 25 years in captivity. While the venom of a few species is dangerous to humans, scientists are now researching the use of spider venom in medicine and as non-polluting pesticides. Spider silk provides a combination of lightness, strength, and elasticity that is superior to that of synthetic materials, and spider silk genes have been inserted into mammals and plants to see if these can be used as silk factories. As a result of their wide range of behaviors, spiders have become common symbols in art and mythology, symbolizing various combinations of patience, cruelty, and creative powers. An irrational fear of spiders is called arachnophobia. Etymology. The word spider derives from Proto-Germanic spintron, literally spinner, a reference to how spiders make their webs, from the Proto-Indo-European root *spen, to draw, stretch, spin. Description. Body plan. Spiders are chelicerates and therefore arthropods. As arthropods they have segmented bodies with jointed limbs, all covered in a cuticle made of chitin and proteins, heads that are composed of several segments that fuse during the development of the embryo. Being chelicerates, their bodies consist of two tagmata sets of segments that serve similar functions. The foremost one, called the cephalothorax or prosoma, is a complete fusion of the segments that in an insect would form two separate tagmata. The head and thorax, the rear tagma, is called the abdomen or opisthosoma. In spiders, the cephalothorax and abdomen are connected by a small cylindrical section, the pedicel. The pattern of segment fusion that forms chelicerates' heads is unique among arthropods, and what would normally be the first head segment disappears at an early stage of development, so that chelicerates lack the antennae typical of most arthropods. In fact, chelicerates' only appendages ahead of the mouth are a pair of chelicerae, and they lack anything that would function directly as jaws. The first appendages behind the mouth are called pedipalps and serve different functions within different groups of chelicerates. Spiders and scorpions are members of one chelicerate group, the arachnids. Scorpions' chelicerae have 3 sections and are used in feeding. Spiders' chelicerae have 2 sections and terminate in fangs that are generally venomous, and fold away behind the upper sections while not in use. The upper sections generally have thick beards that filter solid lumps out of their food, as spiders can take only liquid food. Scorpions' pedipalps generally form large claws for capturing prey, while those of spiders are fairly small appendages whose bases also act as an extension of the mouth. In addition, those of male spiders have enlarged last sections used for sperm transfer. In spiders, the cephalothorax and abdomen are joined by a small, cylindrical pedicel, which enables the abdomen to move independently when producing silk. The upper surface of the cephalothorax is covered by a single, convex carapace, while the underside is covered by 2 rather flat plates. The abdomen is soft and egg-shaped. It shows no sign of segmentation, except that the primitive Mesotheli, whose living members are the Liphistidae, have segmented plates on the upper surface. Circulation and respiration. Like other arthropods, spiders are coelomates in which the coelom is reduced to small areas around the reproductive and excretory systems. Its place is largely taken by a hemicole, a cavity that runs most of the length of the body and through which blood flows. The heart is a tube in the upper part of the body, with a few ostia that act as non-return valves, allowing blood to enter the heart from the hemicole but prevent it from leaving before it reaches the front end. However, in spiders, it occupies only the upper part of the abdomen, and blood is discharged into the hemicole by one artery that opens at the rear end of the abdomen and by branching arteries that pass through the pedicle and open into several parts of the cephalothorax. Hence, spiders have open circulatory systems. The blood of many spiders that have book lungs contains the respiratory pigment hemocyanin to make oxygen transport more efficient. Spiders have developed several different respiratory anatomies based on book lungs, a tracheal system, or both. Mygalomorph and Mesothelies spiders have two pairs of book lungs filled with hemolymph, where openings on the ventral surface of the abdomen allow air to enter and diffuse oxygen. This is also the case for some basal-arenomorph spiders like the family Hypochilidae, but the remaining members of this group have just the anterior pair of book lungs intact, while the posterior pair of breathing organs are partly or fully modified into tracheae, through which oxygen is diffused into the hemolymph or directly to the tissue and organs. The tracheal system has most likely evolved in small ancestors to help resist desiccation. The trachea were originally connected to the surroundings through a pair of openings called spiracles, but in the majority of spiders this pair of spiracles has fused into a single one in the middle and moved backwards close to the spinnerets. Spiders that have tracheae generally have higher metabolic rates and better water conservation. Spiders are ectotherms, so environmental temperatures affect their activity. Feeding, digestion, and excretion. Uniquely among chelicerates, the final sections of spiders' chelicerae are fangs, and the great majority of spiders can use them to inject venom into prey from venom glands in the roots of the chelicerae. The families Uloboridae and Holarchidae and some Lithostidae spiders have lost their venom glands and kill their prey with silk instead. Like most arachnids, including scorpions, spiders have a narrow gut that can only cope with liquid food and two sets of filters to keep solids out. They use one of two different systems of external digestion. Some pump digestive enzymes from the midgut into the prey and then suck the liquefied tissues of the prey into the gut, eventually leaving behind the empty husk of the prey. Others grind the prey to pulp using the chelicerae and the bases of the pedipalps, while flooding it with enzymes. In these species, the chelicerae and the bases of the pedipalps form a prioral cavity that holds the food they are processing. The stomach in the cephalothorax acts as a pump that sends the food deeper into the digestive system. The midgut bears many digestive ceca compartments with no other exit that extract nutrients from the food. Most are in the abdomen, which is dominated by the digestive system, but a few are found in the cephalothorax. Most spiders convert nitrogenous waste products into uric acid, which can be excreted as a dry material. Malpighian tubules, little tubes, extract these wastes from the blood in the hemacole and dump them into the cloacal chamber, from which they are expelled through the anus. Production of uric acid and its removal via Malpighian tubules are a water-conserving feature that has evolved independently in several arthropod lineages that can live far away from water. For example, the tubules of insects and arachnids develop from completely different parts of the embryo. However, a few primitive spiders, the suborder Mesotheli and infraorder Mygalomorphae, retain the ancestral arthropod Nephridia little kidneys, which use large amounts of water to excrete nitrogenous waste products as ammonia. Central nervous system. The basic arthropod central nervous system consists of a pair of nerve cords running below the gut, with paired ganglia as local control centers in all segments, a brain formed by fusion of the ganglia for the head segments ahead of and behind the mouth, so that the esophagus is encircled by this conglomeration of ganglia. Except for the primitive Mesotheli, of which the Lithostidae are the sole surviving family, spiders have the much more centralized nervous system that is typical of arachnids. All the ganglia of all segments behind the esophagus are fused, so that the cephalothorax is largely filled with nervous tissue, and there are no ganglia in the abdomen. In the Mesotheli, the ganglia of the abdomen and the rear part of the cephalothorax remain unfused. Despite the relatively small central nervous system, some spiders like Portia exhibit complex behavior. including the ability to use the trial and error approach. Sense organs. Eyes. Spiders have primarily 4 pairs of eyes on the top front area of the cephalothorax, arranged in patterns that vary from one family to another. The principal pair at the front are of the type called pygmid-cup oculi little eyes, which in most arthropods are only capable of detecting the direction from which light is coming using the shadow cast by the walls of the cup. However, in spiders these eyes are capable of forming images. The other pairs, called secondary eyes, are thought to be derived from the compound eyes of the ancestral chelicerates, but no longer have the separate facets typical of compound eyes. Unlike the principal eyes, in many spiders these secondary eyes detect light reflected from a reflective tapetum lucidum, and wolf spiders can be spotted by torchlight reflected from the tapeta. On the other hand, the secondary eyes of jumping spiders have no tapeta. Other differences between the principal and secondary eyes are that the latter have rhabdomers that point away from incoming light, just like invertebrates, while the arrangement is the opposite in the former. The principal eyes are also the only ones with eye muscles, allowing them to move the retina. Having no muscles, the secondary eyes are immobile. The visual acuity of some jumping spiders exceeds by a factor of 10 that of dragonflies, which have by far the best vision among insects. This acuity is achieved by a telephotographic series of lenses, a 4-layer retina, and the ability to swivel the eyes and integrate images from different stages in the scan. The downside is that the scanning and integrating processes are relatively slow. There are spiders with a reduced number of eyes, the most common having 6 eyes. Example, Periegops suturii with a pair of eyes absent on the anterior median line. Other species have 4 eyes, and members of the Gingponiidae family can have as few as 2. Cave-dwelling species have no eyes or possess vestigial eyes incapable of sight. Other senses. As with other arthropods, spiders' cuticles would block out information about the outside world, except that they are penetrated by many sensors or connections from sensors to the nervous system. In fact, spiders and other arthropods have modified their cuticles into elaborate arrays of sensors. Various touch sensors, mostly bristles called setae, respond to different levels of force, from strong contact to very weak air currents. Chemical sensors provide equivalents of taste and smell, often by means of setae. An adult Araneus may have up to 1,000 such chemosensitive setae, most on the tarsi of the first pair of legs. Males have more chemosensitive bristles on their pedipalps than females. They have been shown to be responsive to sex pheromones produced by females, both contact and airborne. The jumping spider Avarcha culisivora uses the scent of blood from mammals and other vertebrates, which is obtained by capturing blood-filled mosquitoes, to attract the opposite sex. Because they are able to tell the sexes apart, it is assumed the blood scent is mixed with pheromones. Spiders also have in the joints of their limbs slit sensili that detect force and vibrations. In web-building spiders, all these mechanical and chemical sensors are more important than the eyes, while the eyes are most important to spiders that hunt actively. Like most arthropods, spiders lack balance and acceleration sensors and rely on their eyes to tell them which way is up. Arthropods propriosters, sensors that report the force exerted by muscles and the degree of bending in the body and joints, are well understood. On the other hand, little is known about what other internal sensors spiders or other arthropods may have. Some spiders use their webs for hearing, where the giant webs function as extended and reconfigurable auditory sensors. Locomotion. Each of the 8 legs of a spider consists of 7 distinct parts. The part closest to and attaching the leg to the cephalothorax is the coxa. The next segment is the short trochanter that works as a hinge for the following long segment, the femur. Next is the spider's knee, the patella, which acts as the hinge for the tibia. The metatarsus is next, and it connects the tibia to the tarsus, which may be thought of as a foot of sorts. The tarsus ends in a claw made up of either 2 or 3 points, depending on the family to which the spider belongs. Although all arthropods use muscles attached to the inside of the exoskeleton to flex their limbs, spiders and a few other groups still use hydraulic pressure to extend them, a system inherited from their pre-arthropod ancestors. The only extensor muscles in spider legs are located in the 3 hip joints bordering the coccip and the trochanter. As a result, a spider with a punctured cephalothorax cannot extend its legs, and the legs of dead spiders curl up. Spiders can generate pressures up to 8 times their resting level to extend their legs, and jumping spiders can jump up to 50 times their own length by suddenly increasing the blood pressure in the 3rd or 4th pair of legs. Although larger spiders use hydraulics to straighten their legs, unlike smaller jumping spiders, they depend on their flexor muscles to generate the propulsive force for their jumps. Most spiders that hunt actively, rather than relying on webs, have dense tufts of fine bristles between the paired claws at the tips of their legs. These tufts, known as scopulae, consist of bristles whose ends are split into as many as 1,000 branches, and enable spiders with scopulae to walk up vertical glass and upside down on ceilings. It appears that scopulae get their grip from contact with extremely thin layers of water on surfaces. Spiders, like most other arachnids, keep at least 4 legs on the surface while walking or running. Silk production. The abdomen has no appendages except those that have been modified to form 1 to 4, usually 3 pairs of short, movable spinnerets, which emit silk. Each spinneret has many spigots, each of which is connected to one silk gland. There are at least 6 types of silk gland, each producing a different type of silk. Silk is mainly composed of a protein very similar to that used in insect silk. It is initially a liquid, and hardens not by exposure to air, but as a result of being drawn out, which changes the internal structure of the protein. It is similar in tensile strength to nylon and biological materials such as chitin, collagen, and cellulose, but is much more elastic. In other words, it can stretch much further before breaking or losing shape. Some spiders have a cribolum, a modified spinneret with up to 40,000 spigots, each of which produces a single very fine fiber. The fibers are pulled out by the calamistrum, a comb-like set of bristles on the jointed tip of the cribolum, and combined into a composite woolly thread that is very effective in snagging the bristles of insects. The earliest spiders had cribella, which produced the first silk capable of capturing insects before spiders developed silk coated with sticky droplets. However, most modern groups of spiders have lost the cribolum. Even species that do not build webs to catch prey use silk in several ways, as wrappers for sperm and for fertilized eggs, as a safety rope for nest building, and as parachutes by the young of some species. Reproduction and life cycle. Spiders reproduce sexually, and fertilization is internal but indirect. In other words, the sperm is not inserted into the female's body by the male's genitals, but by an intermediate stage. Unlike many land-living arthropods, male spiders do not produce ready-made spermatophores packages of sperm, but spin small sperm webs onto which they ejaculate and then transfer the sperm to special syringe-styled structures, palpal bulbs or palpal organs, borne on the tips of the pedipalps of mature males. When a male detects signs of a female nearby, he checks whether she is of the same species and whether she is ready to mate. For example, in species that produce webs or safety ropes, the male can identify the species and sex of these objects by smell. Spiders generally use elaborate courtship rituals to prevent the large females from eating the small males before fertilization, except where the male is so much smaller that he is not worth eating. In web-weaving species, precise patterns of vibrations in the web are a major part of the rituals, while patterns of touches on the female's body are important in many spiders that hunt actively and may hypnotize the female. Gestures and dances by the male are important for jumping spiders, which have excellent eyesight. If courtship is successful, the male injects his sperm from the palpal bulbs into the female via one or two openings on the underside of her abdomen. Female spiders' reproductive tracts are arranged in one of two ways. The ancestral arrangement, haplogyne or non-intelogyne, consists of a single genital opening leading to two seminal receptacles, spermathecae, in which females store sperm. In the more advanced arrangement in telogyne, there are two further openings leading directly to the spermathecae, creating a flow-through system rather than a first-in first-out one. Eggs are as a general rule only fertilized during oviposition when the stored sperm is released from its chamber, rather than in the ovarian cavity. A few exceptions exist, such as Parastetoda tepidarium. In these species the female appears to be able to activate the dormant sperm before oviposition, allowing them to migrate to the ovarian cavity where fertilization occurs. The only known example of direct fertilization between male and female is an Israeli spider, Harpactia tsaidistica, which has evolved traumatic insemination. In this species the male will penetrate its pedipalps through the female's body wall and inject his sperm directly into her ovaries, where the embryos inside the fertilized eggs will start to develop before being laid. Males of the genus Ptidarin amputate one of their palps before maturation and enter adult life with one palp only. The palps are 20% of the male's body mass in this species, and detaching one of the two improves mobility. In the Yemeni species Ptidarin argo, the remaining palp is then torn off by the female. The separated palp remains attached to the female's epigyne for about 4 hours and apparently continues to function independently. In the meantime, the female feeds on the palpless male. In over 60% of cases, the female of the Australian redback spider kills and eats the male after it inserts its second palp into the female's genital opening. In fact, the males cooperate by trying to impale themselves on the female's fangs. Observation shows that most male redbacks never get an opportunity to mate, and the lucky ones increase the likely number of offspring by ensuring that the females are well fed. However, males of most species survive a few matings, limited mainly by their short lifespans. Some even live for a while in their mates' webs. Females lay up to 3,000 eggs in one or more silk egg sacs, which maintain a fairly constant humidity level. In some species, the females die afterwards, but females of other species protect the sacs by attaching them to their webs, hiding them in nests, carrying them in the chelicerae or attaching them to the spinnerets and dragging them along. Baby spiders pass all their larval stages inside the egg sac and emerge as spiderlings, very small and sexually immature, but similar in shape to adults. Some spiders care for their young. For example, a wolf spider's brood clings to rough bristles on the mother's back, and females of some species respond to the bedding behavior of their young by giving them their prey, provided it is no longer struggling or even regurgitate food. Like other arthropods, spiders have to molt to grow as their cuticle skin cannot stretch. In some species males mate with newly molted females, which are too weak to be dangerous to the males. Most spiders live for only 1 to 2 years, although some tarantulas can live in captivity for over 20 years, and an Australian female trapdoor spider was documented to have lived in the wild for 43 years, dying of a parasitic wasp attack. Coloration. Only 3 classes of pigment amachrones, bilins and guanine, have been identified in spiders, although other pigments have been detected but not yet characterized. Melanins, carotenoids, and pterins, very common in other animals, are apparently absent. In some species, the exocuticle of the legs and prosoma is modified by a tanning process, resulting in a brown coloration. Bilins are found, for example, in Micromatovirusins, resulting in its green color. Guanine is responsible for the white markings of the European garden spider Araneus diadematus. It is in many species accumulated in specialized cells called guanocytes. In genera such as Tetradnatha, Leucoch, Argyrodes, or Theridiosoma, guanine creates their silvery appearance. While guanine is originally an end product of protein metabolism, its excretion can be blocked in spiders, leading to an increase in its storage. Structural colors occur in some species, which are the result of the diffraction, scattering, or interference of light, for example by modified setae or scales. The white prosoma of Argiope results from bristles reflecting the light. Lycosa and Joseph both have areas of modified cuticle that act as light reflectors. The peacock spiders of Australia, genus Meratus, are notable for their bright structural colors in the males. While in many spiders color is fixed throughout their lifespan, in some groups color may be variable in response to environmental and internal conditions. Choice of prey may be able to alter the color of spiders. For example, the abdomen of Theridion grallator will become orange if the spider ingests certain species of Diptera and adult Lepidoptera, but if it consumes Homoptera or larval Lepidoptera, then the abdomen becomes green. Environmentally induced color changes may be morphological, occurring over several days, or physiological, occurring near instantly. Morphological changes require pigment synthesis and degradation. In contrast to this, physiological changes occur by changing the position of pigment-containing cells. An example of morphological color changes is background matching. Miss Eumena vatia, for instance, can change its body color to match the substrate it lives on, which makes it more difficult to be detected by prey. An example of physiological color change is observed in Sitophora cicatrosa, which can change its body color from white to brown near instantly. Ecology and behavior. Non-predatory feeding. Although spiders are generally regarded as predatory, the jumping spider, Bathera kaplingi, gets over 90% of its food from fairly solid plant material produced by acacias as part of a mutually beneficial relationship with a species of ant. Juveniles of some spiders in the families Anaphenidae, Cornidae, Clubionidae, Tomasidae, and Salticidae feed on plant nectar. Laboratory studies show that they do so deliberately and over extended periods, and periodically clean themselves while feeding. These spiders also prefer sugar solutions to plain water, which indicates that they are seeking nutrients. Since many spiders are nocturnal, the extent of nectar consumption by spiders may have been underestimated. Nectar contains amino acids, lipids, vitamins, and minerals in addition to sugars, and studies have shown that other spider species live longer when nectar is available. Feeding on nectar avoids the risks of struggles with prey and the costs of producing venom and digestive enzymes. Various species are known to feed on dead arthropods, scavenging, web silk, and their own shed exoskeletons. Pollen caught in webs may also be eaten, and studies have shown that young spiders have a better chance of survival if they have the opportunity to eat pollen. In captivity, several spider species are also known to feed on bananas, marmalade, milk, egg yolk, and sausages. Capturing prey. The best known method of prey captured is by means of sticky webs. Varying placement of webs allows different species of spider to trap different insects in the same area. For example, flat horizontal webs trap insects that fly up from vegetation underneath, while flat vertical webs trap insects in horizontal flight. Web-building spiders have poor vision, but are extremely sensitive to vibrations. Females of the water spider Argyrontoquatica build underwater diving bell webs that they fill with air and use for digesting prey, molting, mating, and raising offspring. They live almost entirely within the bells, darting out to catch prey animals that touch the bell or the threads that anchor it. A few spiders use the surfaces of lakes and ponds as webs, detecting trapped insects by the vibrations that these cause while struggling. Net-casting spiders weave only small webs, but then manipulate them to trap prey. Those of the genus Hyptiotes and the family Theridiosomatidae stretch their webs and then release them when prey strike them, but do not actively move their webs. Those of the family Danapidae weave even smaller webs, hold them outstretched between their first two pairs of legs, and lunge and push the webs as much as twice their own body length to trap prey, and this move may increase the web's area by a factor of up to 10. Experiments have shown that Deinopes spinosus has two different techniques for trapping prey: backwards strikes to catch flying insects, whose vibrations it detects, and forward strikes to catch ground-walking prey that it sees. These two techniques have also been observed in other Dainipids. Walking insects form most of the prey of most Dainipids, but one population of Deinopes subrufa appears to live mainly on tipulid flies that they catch with the backwards strike. Mature female bullous spiders of the genus Mastophora build webs that consist of only a single trapeze line, which they patrol. They also construct a bullous made of a single thread, tipped with a large ball of very wet sticky silk. They emit chemicals that resemble the pheromones of moths, and then swing the bullets at the moths. Although they miss on about 50% of strikes, they catch about the same weight of insects per night as web-weaving spiders of similar size. The spiders eat the bullets if they have not made a kill in about 30 minutes, rest for a while, and then make new bullets. Juveniles and adult males are much smaller and do not make bolas. Instead, they release different pheromones that attract moth flies, and catch them with their front pairs of legs. The primitive Liphistidae, the trapdoor spiders of the family Ctenizidae, and many tarantulas are ambush predators that lurk in burrows, often closed by trapdoors and often surrounded by networks of silk threads that alert these spiders to the presence of prey. Other ambush predators do without such aids, including many crab spiders and a few species that prey on bees, which see ultraviolet, can adjust their ultraviolet reflectance to match the flowers in which they are lurking. Wolf spiders, jumping spiders, fishing spiders, and some crab spiders capture prey by chasing it and rely mainly on vision to locate prey. Some jumping spiders of the genus Portia hunt other spiders in ways that seem intelligent, outflanking their victims or luring them from their webs. Laboratory studies show that Portia's instinctive tactics are only starting points for trial and error approach from which these spiders learn very quickly how to overcome new prey species. However, they seem to be relatively slow thinkers, which is not surprising, as their brains are vastly smaller than those of mammalian predators. Ant-mimicking spiders face several challenges. They generally develop slimmer abdomens and false waists in the cephalothorax. To mimic the three distinct regions stigmata of an ant's body, they wave the first pair of legs in front of their heads to mimic antennae which spiders lack, and to conceal the fact that they have 8 legs rather than 6, they develop large color patches round one pair of eyes to disguise the fact that they generally have 8 simple eyes, while ants have 2 compound eyes. They cover their bodies with reflective bristles to resemble the shiny bodies of ants. In some spider species, males and females mimic different ant species, as female spiders are usually much larger than males. Ant-mimicking spiders also modify their behavior to resemble that of the target species of ant. For example, many adopt a zigzag pattern of movement. Ant-mimicking jumping spiders avoid jumping, and spiders of the genus Cynomycena walk on the outer edges of leaves in the same way as Pseudomyimex. Ant mimicry in many spiders and other arthropods may be for protection from predators that hunt by sight, including birds, lizards, and spiders. However, several ant-mimicking spiders prey either on ants or on the ants' livestock, such as aphids. When at rest, the ant-mimicking crab spider Amisia does not closely resemble Ocophila, but while hunting it imitates the behavior of a dying ant to attract worker ants. After a kill, Some add mimicking spiders hold their victims between themselves and large groups of ants to avoid being attacked. Defense. There is strong evidence that spiders' coloration is camouflage that helps them to evade their major predators, birds and parasitic wasps, both of which have good color vision. Many spider species are colored so as to merge with their most common backgrounds, and some have disruptive coloration, stripes and blotches that break up their outlines. In a few species, such as the Hawaiian happy-faced spider, Theridion grallator, several coloration schemes are present in a ratio that appears to remain constant, and this may make it more difficult for predators to recognize the species. Most spiders are insufficiently dangerous or unpleasant tasting for warning coloration to offer much benefit. However, a few species with powerful venom, large jaws, or irritant bristles have patches of warning colors, and some actively display these colors when threatened. Many of the family Theraposidae, which includes tarantulas and baboon spiders, have urticating hairs on their abdomens and use their legs to flick them at attackers. These bristles are fine setae bristles with fragile bases and a row of barbs on the tip. The barbs cause intense irritation, but there is no evidence that they carry any kind of venom. A few defend themselves against wasps by including networks of very robust threads in their webs, giving the spider time to flee while the wasps are struggling with the obstacles. The golden wheeling spider, Carparactin aureiflava of the Namibian desert, escapes parasitic wasps by flipping onto its side and cartwheeling down sand dunes. Socialization. A few spider species that build webs live together in large colonies and show social behavior, although not as complex as in social insects. Aelyssimus eximius in the family Theridiidae can form colonies of up to 50,000 individuals. The genus Aelyssimus has a strong tendency towards sociality. All known American species are social, and species in Madagascar are at least somewhat social. Members of other species in the same family, but several different genera have independently developed social behavior. For example, although Theridion nigronulatum belongs to a genus with no other social species, T. nigronulatum builds colonies that may contain several thousand individuals that cooperate in prey capture and share food. Other communal spiders include several Philopponella, species family Uloboridae, Agelena consociata family Agelenidae, and Mallows gregulus family Dictinidae. Social predatory spiders need to defend their prey against kleptoparasites, thieves, and larger colonies are more successful in this. The herbivorous spider Bagheera kaplangi lives in small colonies which help to protect eggs and spiderlings. Even widow spiders genus Latrodus, which are notoriously cannibalistic, have formed small colonies in captivity, sharing webs and feeding together. In experiments, spider species like Steatoda grossa, Latrodus hesperus, and Eredigena agrestes stayed away from Myrmica rubra ant colonies. These ants are predators, and the pheromones they release for communication have a notable deterrent effect on these spider species. Web types. There is no consistent relationship between the classification of spiders and the types of web they build. Species in the same genus may build very similar or significantly different webs. Nor is there much correspondence between spiders' classification and the chemical composition of their silks. Convergent evolution in web construction, in other words, use of similar techniques by remotely related species, is rampant. Orb web designs and the spinning behaviors that produce them are the best understood. The basic radial then spiral sequence visible in orb webs and the sense of direction required to build them may have been inherited from the common ancestors of most spider groups. However, the majority of spiders build non-orb webs. It used to be thought that the sticky orb web was an evolutionary innovation resulting in the diversification of the Orbicularia. Now, however, it appears that non-orb spiders are a subgroup that evolved from orb web spiders and non-orb spiders have over 40% more species and are 4 times as abundant as orb-web spiders. Their greater success may be because specid wasps, which are often the dominant predators of spiders, much prefer to attack spiders that have flat webs. Orb. About half the potential prey that hit orb webs escape. A web has to perform 3 functions: intercepting the prey intersection, absorbing its momentum without breaking, stopping, and trapping the prey by entangling it or sticking to it retention. No single design is best for all prey. For example, wider spacing of lines will increase the web's area and hence its ability to intercept prey, but reduce its stopping power and retention. Closer spacing, larger sticky droplets, and thicker lines would improve retention, but would make it easier for potential prey to see and avoid the web, at least during the day. However, there are no consistent differences between orb webs built for use during the day and those built for use at night. In fact, there is no simple relationship between orb web design features and the prey they capture, as each orb-weaving species takes a wide range of prey. The hubs of orb webs, where the spiders lurk, are usually above the center, as the spiders can move downwards faster than upwards. If there is an obvious direction in which the spider can retreat to avoid its own predators, the hub is usually offset towards that direction. Horizontal orb webs are fairly common, despite being less effective at intercepting and retaining prey and more vulnerable to damage by rain and falling debris. Various researchers have suggested that horizontal webs offer compensating advantages such as reduced vulnerability to wind damage, reduced visibility to prey flying upwards because of the backlighting from the sky, enabling oscillations to catch insects in slow horizontal flight. However, there is no single explanation for the common use of horizontal orb webs. Spiders often attach highly visible silk bands, called decorations or stabilimenta, to their webs. Field research suggests that webs with more decorative bands captured more prey per hour. However, a laboratory study showed that spiders reduce the building of these decorations if they sense the presence of predators. There are several unusual variants of orb web, many of them convergently evolved, including attachment of lines to the surface of water, possibly to trap insects in or on the surface, webs with twigs through their centers, possibly to hide the spiders from predators, ladder-like webs that appear most effective in catching moths. However, the significance of many variations is unclear. The orb-weaving species Zygiella X notata, for example, is known for its characteristic missing sector orb web. The missing sector contains a signal thread used to detect prey vibrations on the female's web. In 1973, Skylab 3 took 2 orb-webbed spiders into space to test their web-spinning capabilities in zero gravity. At first, both produced rather sloppy webs, but they adapted quickly. Cobweb. Members of the family Theridiidae weave irregular, tangled, 3-dimensional webs, popularly known as cobwebs. There seems to be an evolutionary trend towards a reduction in the amount of sticky silk used, leading to its total absence in some species. The construction of cobwebs is less stereotyped than that of orb webs and may take several days. Other. The Linniphidae generally make horizontal but uneven sheets with tangles of stopping threads above. Insects that hit the stopping threads fall onto the sheet or are shaken onto it by the spider, and are held by sticky threads on the sheet until the spider can attack from below. Web design in zero gravity. Many experiments have been conducted to study the effect of zero gravity on the design of spider webs. In late 2020, reports of recent experiments were published that indicated that although web design was affected adversely in zero-gravity conditions, having access to a light source could orient spiders and enable them to build their normally shaped webs under such conditions. Evolution. Fossil record. Although the fossil record of spiders is considered poor, almost 1,000 species have been described from fossils. Because spiders' bodies are quite soft, the vast majority of fossil spiders have been found preserved in amber. The oldest known amber that contains fossil arthropods dates from 130 million years ago in the early Cretaceous period. In addition to preserving spiders' anatomy in very fine detail, pieces of amber show spiders mating, killing prey, producing silk, and possibly caring for their young. In a few cases, amber has preserved spiders' egg sacs and webs, occasionally with prey attached. The oldest fossil web found so far is 100 million years old. Earlier spider fossils come from a few Lagerstätten, places where conditions were exceptionally suited to preserving fairly soft tissues. The oldest known exclusively terrestrial arachnid is the trigonotarbid Paleotarbis jerami from about 420 million years ago in the Silurian period, and had a triangular cephalothorax and segmented abdomen, as well as 8 legs and a pair of pedipalps. Atercopus fimbriunguis from 386 million years ago in the Devonian period bears the earliest known silk-producing spigots and was therefore hailed as a spider at the time of its discovery. However, these spigots may have been mounted on the underside of the abdomen rather than on spinnerets, which are modified appendages, and whose mobility is important in the building of webs. Hence, Atercopus and the similar Permian arachnid Permaract may not have been true spiders, and probably used silk for lining nests or producing egg cases rather than for building webs. The largest known fossil spider as of 2011 is the Aranidnephila jurassica from about 165 million years ago, recorded from Daehuogo, Inner Mongolia in China. Its body length is almost 25 mm, i.e., almost 1 inch. Several Carboniferous spiders were members of the Mesophylae, a primitive group now represented only by the Liphistidae. The mesothelid Paleothelmid Soensis, from the late Carboniferous over 299 million years ago, had 5 spinnerets. Although the Permian period 299 to 251 million years ago saw a rapid diversification of flying insects, there are very few fossil spiders from this period. The main groups of modern spiders, Mygalomorphae and Araenomorphae, first appear in the Triassic well before 200 million years ago. Some Triassic mygalomorphs appear to be members of the family Hexathelidae, whose modern members include the notorious Sydney funnel-web spider, and their spinnerets appear adapted for building funnel-shaped webs to catch jumping insects. Araenomorphae account for the great majority of modern spiders, including those that weave the familiar orc-shaped webs. The Jurassic and Cretaceous periods provide a large number of fossil spiders, including representatives of many modern families. External relationships. The spiders Araeni are monophyletic, i.e., a clade consisting of a last common ancestor and all of its descendants. There has been debate about what their closest evolutionary relatives are, and how all of these evolved from the ancestral chelicerates, which were marine animals. This 2019 cladogram illustrates the spiders' phylogenetic relationships. Arachnids lack some features of other chelicerates, including backward-pointing mouths and gnathobases jaw bases at the bases of their legs. Both of these features are part of the ancestral arthropod feeding system. Instead, they have mouths that point forwards and downwards, and all have some means of breathing air. Spiders Araeni are distinguished from other arachnid groups by several characteristics, including spinnerets and, in males, pedipalps that are specially adapted for sperm transfer. Internal relationships. The cladogram shows the relation among spider suborders and families. Taxonomy. The order name Araeni derives from Latin Arania, borrowing ancient Greek letter 1F00 rho alpha tenuita arachn from letter 1F00 rho alpha tenuita sigma arachns. Spiders are divided into 2 suborders, Mesotheli and Opisthotheli, of which the latter contains two inforidors, Mygallomorphae and Araenomorphae. Some 50,356 living species of spiders order Araeni have been identified, grouped into 132 families and 4,280 genera by arachnologists in 2022. Mesothelae. The only living members of the primitive Mesothelae are the family Liphistidae, found only in Southeast Asia, China, and Japan. Most of the Liphistidae construct silk-lined burrows with thin trapdoors, although some species of the genus Liphistius build camouflaged silk tubes with a second trapdoor as an emergency exit. Members of the genus Liphistius run silk tripwires outwards from their tunnels to help them detect approaching prey, while those of the genus Heptaphila do not and instead rely on their built-in vibration sensors. Spiders of the genus Heptaphila have no venom glands, although they do have venom gland outlets on the fang tip. The extinct families Arthrolechacidae, found in Carboniferous and Permian rocks, and Arthromegallidae, so far found only in Carboniferous rocks, have been classified as members of the Mesothelae. Mygallomorphae. The Mygallomorphae, which first appeared in the Triassic period, are generally heavily built and hairy with large, robust chelicerae and fangs. Technically, spiders do not have true hairs, but rather setae. Well-known examples include tarantulas, C-tenizid trapdoor spiders, and the Australasian funnel-web spiders. Most spend the majority of their time in burrows, and some run silk tripwires out from these, but a few build webs to capture prey. However, Mygallomorphs cannot produce the piriform silk that the Araenomorphae use as an instant adhesive to glue silk to surfaces or to other strands of silk, and this makes web construction more difficult for Mygallomorphs. Since Mygallomorphs rarely balloon by using air currents for transport, their populations often form clumps. In addition to arthropods, some Mygallomorphs are known to prey on frogs, small mammals, lizards, snakes, snails, and small birds. Araenomorphae. In addition to accounting for over 90% of spider species, the Araenomorphae, also known as the true spiders, include orb-web spiders, the cursorial wolf spiders, and jumping spiders, as well as the only known herbivorous spider, the Hieracoplingi. They are distinguished by having fangs that oppose each other and cross in a pinching action, in contrast to the Mygallomorphae, which have fangs that are nearly parallel in alignment. Human interaction. Media coverage and misconceptions. Information about spiders in the media is often emphasizing how dangerous and unpleasant they are. A recent study investigated the global spread of misinformation on spiders using a high-resolution global database of online newspaper articles on spider-human interactions. These reports covered stories of spider-human encounters and bites published from 2010 to 2020. The study found that 47% of articles contained errors and 43% were sensationalist. Bites. Although spiders are widely feared, only a few species are dangerous to people. Spiders will only bite humans in self-defense, and few produce worse effects than a mosquito bite or bee sting. Most of those with medically serious bites, such as recluse spiders genus Loxosceles and widow spiders genus Latrodictis, would rather flee and bite only when trapped, although this can easily arise by accident. The defensive tactics of Australian funnel-web spiders family Atracidae include fang display. Their venom, although they rarely inject much, has resulted in 13 attributed human deaths over 50 years. They have been deemed to be the world's most dangerous spiders on clinical and venom toxicity grounds, though this claim has also been attributed to the Brazilian wandering spider genus Phonutria. There were about 100 reliably reported deaths from spider bites in the 20th century, compared to about 1,500 from jellyfish stings. Many alleged cases of spider bites may represent incorrect diagnoses, which would make it more difficult to check the effectiveness of treatments for genuine bites. A review published in 2016 agreed with this conclusion, showing that 78% of 134 published medical case studies of supposed spider bites did not meet the necessary criteria for a spider bite to be verified. In the case of the two genera with the highest reported number of bites, Loxosceles and Latrodus, spider bites were not verified in over 90% of the reports. Even when verification had occurred, details of the treatment and its effects were often lacking. Silp. Because spider silp is both light and very strong, attempts are being made to produce it in goat's milk and in the leaves of plants by means of genetic engineering. Arachnophobia. Arachnophobia is a specific phobia. It is the abnormal fear of spiders or anything reminiscent of spiders, such as webs or spider-like shapes. It is one of the most common specific phobias, and some statistics show that 50% of women and 10% of men show symptoms. It may be an exaggerated form of an instinctive response that helped early humans to survive, or a cultural phenomenon that is most common in predominantly European societies. As food. Spiders are used as food. Cooked tarantulas are considered a delicacy in Cambodia, and by the Paira Indians of southern Venezuela, provided the highly irritant bristles, the spider's main defense system, are removed first. Spiders in culture. Spiders have been the focus of stories and mythologies of various cultures for centuries. Atu, the ancient Sumerian goddess of weaving, was envisioned as a spider spinning her web. According to her main myth, she resisted her father Enki's sexual advances by ensconcing herself in her web, but let him in after he promised her fresh produce as a marriage gift, thereby allowing him to intoxicate her with beer and rape her. Enki's wife Ninhursag heard Atu's screams and rescued her, removing Enki's semen from her vagina and planting it in the ground to produce 8 previously non-existent plants. In a story told by the Roman poet Ovid in his Metamorphoses, Arachne was a Lydian girl who challenged the goddess Athena to a weaving contest. Arachne won, but Athena destroyed her tapestry out of jealousy, causing Arachne to hang herself. In an act of mercy, Athena brought Arachne back to life as the first spider. Stories about the trickster spider Anansi are prominent in the folktales of West Africa and the Caribbean. In some cultures, spiders have symbolized patience due to their hunting technique of setting webs and waiting for prey, as well as mischief and malice due to their venomous bites. The Italian tarantella is a dance to rid the young woman of the lustful effects of a spider bite. Web spinning also caused the association of the spider with creation myths, as they seem to have the ability to produce their own worlds. Dream catchers are depictions of spider webs. The Mach people of ancient Peru worshipped nature. They placed emphasis on animals and often depicted spiders in their art. Citations. General and cited references.
Thank you for watching. Consider supporting the channel, leave a like and subscribe.